Hello, everyone, and welcome back to Lady Scientist Podcast. I'm your host, Jocelyn Pearl, and today we have a great interview for you. I chat with Lexi Walls. She's a postdoc researcher at the University of Washington, and she actually kind of predicted the global pandemic. She was working on coronaviruses and spike proteins before we were seeing them all over the news. We talk about that experience and more in this episode. If you can, subscribe to our channel, leave us a like, comment. Also want to give a shout out to Kendall Investor Relations. They're our sponsor for this episode. We really appreciate you supporting Lady Scientist Podcast. And if you're a biotech entrepreneur out there looking for some help speaking with investors, please reach out to Kendall Investor Relations. I'll put the link below. Thanks so much for listening, guys, and I hope you enjoy this interview with Lexi Walls. Well, I'm really glad you reached out and we get to chat today about your research because I've read a little bit from some of the articles that you've been in, but I think you've had this really interesting experience with regards to the global pandemic and the fact that you have been studying coronaviruses for a while now. So I guess before I even jump into the questions, I just want to introduce Lexi Walls, who studied coronaviruses for her PhD and is now working on vaccine and therapeutic development at the University of Washington. I'm really excited to ask her all of my coronavirus questions and get into some of the nerdy biology there. So welcome, Lexi.
Thanks for having me.
So you defended your dissertation right on the eve of the global pandemic in December 2019. Can you walk us through what that experience was like and, maybe some of the first things that you heard about with regards to COVID and what was happening and how, what that was like?
Yeah, honestly, it's not that long ago, but it feels like centuries ago, given how much we've learned and changed and done in the last year and a half. But yeah, so we started hearing, whispers that there was, some infections that beginning to mid-December 2019, but nothing had been confirmed, nothing had been identified. And my boss and I were like, what if it's a coronavirus? Just wondering. Because it was respiratory, we were like, okay, it could be influenza, it could be coronavirus, it could be a paramyxovirus, just going through what we knew. And when we finally found out that it was a coronavirus, it was surreal because, this had happened twice in the past 20 years, first in 2002 with SARS, and second in 2012 with MERS. And then for it to be happening, as I'm wrapping up, as I'm trying to, leave, figure out my next steps, and then boom, we're in it. It was surreal, and it still is surreal most days, actually.
I can imagine. How did you get your start studying these types of viruses? Was there, were you kind of deciding between different types of viruses and then settled on this one in particular? And what led to that decision?
So it sounds kind of crazy to say now, but I started studying coronaviruses in 2015. And at that time, we didn't know very much about anything coronavirus related. There had been a very small, tight-knit field that had been working really steadily to, make gains and understand these viruses. But when I started working, there was no structure or no real vision of what the spike protein, which as many of you have heard about, which is on the exterior of the virus, really no vision of what this looked like. And so that's kind of where I got excited and stepped in. I was like, this is a place I can contribute that, there's a clear gaping hole of knowledge. Let's see what we can do. And so we had kind of this knowledge gap, and also this new technique called cryo-electron microscopy was really, exploding and coming to the forefront. And so it was kind of this perfect merging of, cryo-EM and really an understudied field that got me excited and interested. And I'm still here.
Wow. Are there other major players, as far as this scientific field that are, that were working on coronaviruses at that point in time? were there collaborators that you were talking to at other organizations, or, was a lot of your research really independent?
No, one thing that's been really fun about my PhD and beyond is we've had tons of collaborators kind of all over the world, which has been really exciting and nice that, I am in a structural biology lab, and we've morphed into, a virology, a vaccinology lab, but we really brought the structural biology expertise, and we collaborated with a ton of other groups who brought the virology and the biology really expertise that we learned from them. And without this merging of all of these expertises, we would not be where we are today, for sure.
Fascinating. So you had a paper, right, that kind of summarized a lot of your academic research from your PhD. When did that come out? And how did that coincide with like the global pandemic?
Yeah, so I was really lucky. I published quite a bit during my PhD. So during my second year is when we published the first structure of a coronavirus spike protein. And basically since then, we've kind of consistently been publishing, first structure of a coronavirus spike protein in one conformation. And then we really tried to understand what it would look like in a different conformation. And we've slowly been building this knowledge arsenal, basically building up to 2019. And so right before, when I defended, I had this whole presentation where I was like, coronaviruses, they have pandemic potential. This is why I think they have pandemic potential. This is why I think we need a vaccine or we need a therapeutic, because before 2020, there was no vaccine for any coronavirus. SARS-2 is not the first coronavirus that infects humans, and we had nothing ready. But luckily, the scientific world was ready. So
why do you think that was? I mean, because aren't there some, aside from the type of coronaviruses that cause respiratory illness, aren't there other coronaviruses circulating? And like, because I'm not in the vaccine space, so I'm just curious why we didn't necessarily have a vaccine yet for something like that.
Yeah, I think there's a lot of ways we could answer that question. I think one of them is, really until 2002, when severe acute respiratory syndrome, or SARS, the original SARS, was discovered and was shown to be, really deadly in humans. It has a 10% fatality rate, but it was only about 8,000 infections worldwide. It was able to be contained, and so, after about a year and a half, people kind of dropped off. Research picked up because everyone was like, what is this virus? What is this virus family? Let's learn something about it. But unfortunately, funding and interest is really driven by, need. And it's always been known that there are coronaviruses that cause common colds. And, growing up, you're always like, why isn't there a vaccine against the common cold? Like, that would be so great. But the truth is, like, the common cold, while it affects all of us and it sucks and it's terrible, there's, not a driving, desperate need for this type of vaccine. And so I think the money and the push just wasn't there because, now that we've gone through the whole process, it takes a lot of people, a lot of money, and a lot of dedication to get even just one vaccine through the door. So yeah, I think it was a number of factors.
Yeah, you mentioned that your dissertation work predicted that these types of viruses could cause a pandemic. What were the factors that led you to make that type of statement?
Yeah, so there's coronaviruses that infect humans, but there's also coronaviruses that infect a whole host of different animals. And, I hadn't done very much of this work, but a lot of our collaborators and colleagues in the field had shown, it only takes a few mutations to take a coronavirus that's purely able to infect bats, for example. If you mutate the spike protein in 1, 2, 3 locations, in cell culture, you can get that to infect human cells. And so that really suggests that, these viruses are poised to jump from species to species, and it just takes time and maybe even intersection of these species for the virus to have this opportunity. They're really, 3 mutations is not a lot. Like, they are ready. And we all had known this for a while, and unfortunately we all are living it now.
Yeah. So people were working on, like, what was that happening, while you were getting your PhD, like that type of work, and you were familiar with that?
Absolutely.
Research. So they're engineering these viruses to be able to infect human cells.
They're not engineering them. They're trying to figure out, why are they infecting only bats? Why are they infecting only humans? What is the molecular determinant that causes, this to be a bat-specific virus, this to be a human-specific virus? And you don't have to do this work with viruses. We work with pseudoviruses all the time, so there's nothing infectious about this sample. It's literally just the spike protein on a backbone of a cell. So it's not, you don't even need the whole virus to understand what allows this spike protein to bind to a cell, to enter a cell. Those are kind of the basic questions that the field work trying to answer.
I see. And from like a safety perspective, that type of research, because for instance, I work with viruses, but from a therapeutic perspective and engineering cells to overcome particular disease state. And a lot of that work is in what we call biosafety level 2 type of facilities. Is the research that you're talking about, is that also considered like biosafety level 2, or is it?
Yeah, because it's not, like I said, it's not even a virus. It's literally just, we have a single protein that's associated with coronaviruses, the spike, and we say, okay, we know that it can bind bat ACE2. What mutations would it take for it to bind human ACE2? And you're literally just mutating a protein. There's nothing replicative about this at all. Just trying to understand, what does it take to change from species to species?
Sure. And so what you're saying is that the risk of a pandemic from these types of viruses really comes down to the fact that it wouldn't take that many mutations for it to be potentially really transmissible or infectious. I'm not sure like what the right word is there, but is that the gist of what you're saying?
Yes, and then I also have 2 other reasons.
Okay.
So that's one reason. Another reason is, if you look in the last 20 years, there are only 2 viruses that have emerged novelly for the first time ever. And both of those are coronaviruses. This is, if we just look at history, it's going to repeat itself. So it was kind of a safe bet to say, this has happened twice since 2002. Why can't it happen again? Right? Like, we've seen it happen twice. Why not again? So that's kind of another reason that we were all, predicting that these viruses could jump from animal to human species.
Sure. But what about Ebola?
Yeah, but Ebola's had outbreaks back into the 60s. Yeah. SARS and MERS had never, ever been discovered. Never been known. There's never, in recent history, there's never been known to have a deadly coronavirus. So SARS was the first, MERS the second, and unfortunately SARS-2 the third.
And you said you have a third reason?
Yeah, the third reason is if you go looking in, animal reservoirs in bats, there are hundreds, if not thousands, of coronaviruses there that, we haven't even tapped how many viruses these that are present in bats, but it means that there's really a large species diversity. And like I was saying, it just, when will there be mutations that allow them to stop being bat coronaviruses and move into human or civet or pangolin or camel? We've had all these intermediates, and it just takes unfortunate mutations that allows that to happen.
Interesting. From a species to species transfer. How would you say that, by the way? Because I feel like I'm not saying that correctly.
No, I think you are. Transmission,
Okay.
yeah.
Okay.
Zoonosis is another
Right.
word.
I mean, how, I guess my question is like, it just seems odd that we could get a virus from a pangolin or a camel. I mean, maybe a bat, right? Like they inhabit a lot of like similar areas. Their feces could, maybe there's a bat living in my attic and it's,
Yeah, hopefully
I
not.
don't know. Like, do you have any thoughts on that? Like,
I think, it's happening not just with coronaviruses, right? We hear about avian influenza or swine flu. This is happening with a lot of different, viruses and probably also bacteria as well. And I think it's just, we come into close contact with animals way more than we might realize. People work on farms or go to zoos or anything, or it can happen anytime. And part of the problem is we're in, like, humans are encroaching on animal habitat, and that could be one of the reasons we're seeing so many emerging viruses, is we're really pushing the boundaries and getting closer and closer to a lot of animal reservoirs.
I see. Interesting. So I want to get back to your dissertation work. Were there any kind of standout days for you where, maybe you saw the structure of this protein for the first time, and can you share that experience with us?
Yeah. So typically, I was a biochemist by nature, and I sort of, joined the structural biology lab, and I had no idea what I was doing. I had never used a microscope before. I had never opened the terminal on my computer before to do any sort of computational work. Like, I really had no clue what I was doing. And when you're a biochemist, the ways that we see our proteins are typically, straight lines on a gel. Either it's there or it's not. Either it's at the right size or it's not. Or we have, a functional assay. Like, if your enzyme has activity, you will have a color change, or you will see some sort of readout. And the first time that I put a protein that I purified onto a negative stain grid, it's this tiny circle, and then imaged it in the microscope, this was truly the first time I was seeing my work. It wasn't a test tube, it wasn't a line on a gel. This was the first time I saw what I had done. And that got me, I was doing my rotations, and that was the moment I knew I was joining this lab. I was like, this is cool. Like, I can see the work I'm doing. That was very cool. And then really, I've been very lucky to work, like, my boss, his name's David Wiesler. We've been really lucky that, both of us have had a lot of successes over the last couple of years. But when we got the first paper accepted, he was a brand new PI, I was a brand new grad student, and just going through that whole process for the first time, and then the paper finally being accepted, was a really, really great day. We knew that we had the first structure of a coronavirus spike protein, and it was about to be, out in the world. This was before bioarchive, so we were the only ones who knew about it, and I was so excited to be able to share that.
Wow, that's incredible. And is that the protein, like, is that the structure that we see in a lot of these articles?
Yeah, so this was a while back, but most of the structures that you see are of the spike protein. And early in 2020, we and another group at UT Austin solved the structure of the spike protein, and that's the image you see is the work we were doing.
Very cool. That's really neat. So you're wrapping up your dissertation. This outbreak is being talked about. Had you already decided where you wanted to go for your postdoc, or did that really like change things for you?
It changed everything. So I was finishing up, and like many people, I had a paper that I wanted to finish, and I wanted to defend, but I hadn't quite finished this paper. And so I said, I want to stay 3 to 6 more months I'll finish this paper while I'm applying for, my postdoc, figuring out what the best fit is for me. Because, the last 3 to 6 months of your PhD is kind of terrifying and terrible and stressful. And I just needed some extra time to figure out what was coming next. But, we found out it was a coronavirus early January. And then we found the sequence was released like early to mid January of 2020. And, right away ordered genes, was already planning experiments, and, no one knew at that point what was coming, right? I was like, okay, we're gonna, figure out what the receptor is, figure out what the spike structure is, see if we can get a pseudovirus system working. And then obviously it hasn't stopped since.
Wow.
But even at that point, I was like, okay, this is one more project. I'm still going to do my other project in the background. One more project, we'll get the spike structure out, we're done. I had no idea that this was what was coming.
Right.
And that other paper is still not published, by the way.
Classic. I still have one from grad school that is not out there yet.
Yeah.
So where did the sequence come from? Like the initial sequence?
Yeah. So this was by heroic efforts from a lot of Chinese scientists who basically sequenced the genome for the first time, and they made the decision to share the sequence publicly and openly. And I first found it on Twitter, actually. So someone tweeted, the sequence is here. You can go to the GuyZ, directory and find it. And, literally I was sitting there immediately, start Googling. I'm like, okay, got to get this directory, got to go here, and, just start figuring out what the sequence was. So really it was through knowledge sharing that we all were able to start working on this so fast.
Interesting. It's been fascinating how much information share has been going on over Twitter, like during this time, and almost like more direct communication than I feel like we would have had without that type of platform.
Yeah, Twitter and also bioarchive have changed completely over the course of the pandemic, and I think for the most part I can say for the better.
Yeah.
There are people who are sharing data on Twitter before it even gets to bioarchive. Like, that's how fast we are all as a community sharing our data, because you have to, the whole world is waiting on answers and on what's happening.
Okay, so I'm going to ask you a couple like more in-depth nerdy questions.
Great, I'm here
even though it might not be, depending on who's listening. So you find the sequence, you download it. What is in there? Like I'm less familiar. I mean, most of these viral genomes, how many proteins do they encode?
Yeah, so it depends. When I first downloaded, it's about, 30 kb, so 30,000 base pairs.
So that's pretty big.
So coronaviruses are one of the larger RNA viruses, so they're huge. I'm gonna get the number of proteins wrong, but it's somewhere in the 7 to 10 range.
Okay.
And most of them are non-structural, so they're helper proteins basically. And then the spike, which is really what I focus on. I don't work on the nucleocapsid or any of those other proteins, which have extremely important roles in dampening our immune response or heightening our immune response or giving structure to the virus. But I really work on the spike, which is responsible for getting the virus into our cells, and also is really the main target of our immune system, because it's the protein that's on the exterior of the virus.
Right.
But the spike is really at the end of the genome, and I, went in. I'm also not a genome person, but went in, found where the cutoffs that I expected the spike to be based on other coronaviruses, and then started translating into proteins, started aligning, we had literally no information about this other than it was a coronavirus. I was like, what coronavirus is this spike most similar to? Can I predict what its likely receptor is based on, what does the receptor binding domain look like? What other coronavirus spike does this look like? And, just
which
getting
other
as
ones,
much info.
Which other ones did it look like? Did it match up to?
So it was most similar to SARS, which we had studied and we had been looking at. We had just put a paper out on neutralizing antibody against SARS. And so, our mind starts racing, like, will it have the same receptor as SARS? Will neutralizing antibodies against SARS bind and or neutralize SARS-2? It wasn't named SARS-2 yet, but novel coronavirus 19 is what it was called at the time.
Yeah.
And, all these questions just start racing, and you're trying to identify as much as you can just from the sequence alone and seeing what you can learn.
So did you know early on that ACE2 was the receptor?
So we thought that was likely just because it was similar to SARS, but, we had to go prove it in the lab. And so, luckily we already had ACE2 in the lab. We had cells expressing ACE2 in the lab. And so all we needed was, get the gene, make the protein. Can we see binding interaction? And we did. And we, characterized that binding interaction. And then the next question was, okay, we know it binds. Is this sufficient for, for propagating entry? And so, using those pseudoviruses where it's literally just the spike, and have a cell that has ACE2 and a cell that doesn't have ACE2, and, see what enters. And it turns out we and many, many other groups showed, like, yes, ACE2 is the receptor. And yes, ACE2 is really majority of what you need to get this pseudovirus or this virus or anything into a cell. So, yeah.
Interesting. So I want to ask, because you're clearly someone who has spent a lot of time looking at these sequences and obviously had an early look into the sequence, how do you think about the hypothesis that potentially this was leaked from a laboratory?
Yeah, I think, we're scientists. And so I am personally a fan of the mantra, show me the data. So, I am not ruling anything out, but until I see data that says, yes, this was a lab leak, no, this was not a lab leak, I don't think anyone knows. And I don't think it even matters really. We're here, we're in it. And we just need to find ways to stop the global pandemic as it is right now, unfortunately.
Absolutely.
I do think, like we talked about earlier, this is not the first time that a virus has jumped from animals to humans. And so it's not unlikely that that SARS-2 is no different from SARS or from MERS. And until we see data otherwise, that's what we got to go with. But I think more data is always good. So I'm happy to see the data as it keeps coming out.
Yeah, there was a paper recently that was comparing some of the sequences from different projects that, were studying coronavirus before the pandemic and looking at some of the particular mutations that were present. Have you seen any of that data, or do you have any thoughts on some of those claims?
I haven't looked in detail in the paper because, again, it doesn't change the situation that we're in right now. But I think until we as a whole scientific community can accept, one or the other, it's going to be a hot debate no matter what.
For sure. On that note, you now work on therapeutics and vaccine development. When did you kind of shift from more of the biology side or structural biology to therapeutic or vaccine work? And can you share what that has been like?
Yeah, so kind of towards the end of my PhD, like I said, we had started working on, neutralizing antibodies. And this is kind of, there are multiple directions that you can take therapeutics. One is a preventative, like a vaccine. It hopefully stops you from getting sick in the first place. And then a second is, is there something we can do once you're already sick? And in general, that's what, antibody therapy is if you're already sick, what can we do for you? And then there's kind of a crosstalk between these. If we know that we have a neutralizing antibody or an antibody therapy that works, can we work backwards and say, how do we produce a vaccine that gets us this response? It's both of them talk to each other and both of them learn from each other. And so towards the end of my PhD, I started working, on neutralizing antibodies, and I started thinking about vaccines, and we started even collaborating with Neil King's lab at the UW IPD, kind of towards the end of my PhD, just seeing like, would this work in mice? Like, what can we see? Can we get a good immune response from this type of vaccine? And then, obviously pandemic happened, and, our first thing was, let's learn as much as we can about this virus. And so that's really what our first paper was: structure, receptor, do we have any crosstalk between known neutralizing antibodies from SARS or from other coronaviruses? And then, the pandemic was still happening, and we were sitting there and we were like, well, we are experts in coronavirus, and we work right down the hall from experts in vaccine design. Like, let's merge two awesome fields and let's take our expertise and run with it and see how it goes. And that's really how it started. It started out of necessity. Like, we don't have a vaccine. Can we help? Yes, let's try.
That's awesome. And so what approach did you guys take with the vaccine? Because, many of us are now familiar with the mRNA vaccines. Pfizer and Moderna's vaccine are mRNA based, whereas more traditional vaccines have kind of a different backbone, I'll call it.
Yeah.
So can you talk us through the flavor of the type of vaccines that you guys have been working on?
Yeah, so ours is totally different from both of those, which is fun and exciting, I think. So ours is called a subunit protein vaccine. So no mRNA, no virus, it's literally just protein. And what we've chosen to do is to kind of combine two things. So we've taken a piece of the coronavirus, specifically a piece of the spike protein. And what we've done is we've fused this to a designed protein. And so what I mean by designed protein is the UW Institute for Protein Design, or IPD, as I previously called it. They are trying to build proteins from scratch. So nothing that's been found in nature, and they're trying to say, can we take a sequence and give it and predict and design a three-dimensional shape and function? And so that's really what we've done, is we've fused a piece of the coronavirus spike protein to a designed protein that forms a beautiful, what we call a nanocage. So it's this beautiful 3D structure that's forming kind of a cage, or like a soccer ball. And then on the end of the soccer ball, you have kind of these pieces of spike protein that are floating off of it.
Oh, wow. Okay. So the designed protein forms kind of like a viral capsid, but then it has the spike sticking out.
Exactly. That's what it looks like.
Okay. Very cool. That's neat. So you've tested this now, like where is it in the process? You've tested it in mice or non-human primates?
Yeah. Even further, it is in phase 1-2 clinical trials in humans,
which
is amazing. Yeah,
I didn't know
amazing. That. So yeah, we started in mice, and we basically we started just in the test tube and said, does this behave like a vaccine? Like, do we express the proteins properly? Are they folded? Are they behaving? And that checked. And so then we moved into mice, and we said, okay, if we use this as a vaccine in mice, do mice get a strong immune response? And they did. It blew us out of the water what we were expecting. But, these mice had extremely strong protective immune responses against SARS-2. And so then we moved to non-human primates, and, same thing at every step of the way. This is just done better than we could have dreamed. And so, we've gone from the test tube to mice to non-human primates, and now it's all the way in humans. And it just got approved for phase 3 clinical trials. So we have funding
Wow.
all the way through phase 3. Yeah, it's amazing.
That's huge.
Yeah, it's really exciting.
Congrats. And okay, like again, kind of nerdy question here, but one thing that I think is hard to, tease out from a lot of these trials that are going on, is there any point in the process of engineering these vaccines where you're able to compare them to like an mRNA approach or to another type of vaccine?
Yeah, so at every step of the way, we call it benchmark our vaccine. So, everyone's assays are slightly different. And so what we wanted to do was say, look, this is a brand new type of vaccine. Is it doing better than what's out there, or at the time what was out there? And so when we first tested in mice, we benchmarked against just the spike protein. So not an mRNA vaccine. This wasn't, available or out yet, but just spike protein, which is kind of a common base of, it is the base of the mRNA vaccine. It's just in protein form rather than nucleic acid form. And
Sure.
what we found was, our vaccine was behaving better and was giving a better immune response. So that was kind of our green light to say, okay, based on the standard at the time, this was before the mRNA vaccines, we are doing better. So we're comfortable, moving ahead and pushing this. And then at every step of the way since, there have been a lot of people in clinical trials, and now, a lot of people have been vaccinated, which is amazing. And so we can take, sera or plasma from people who have been immunized with an mRNA vaccine, for example, and benchmark how our vaccine is doing. Or we can benchmark against, if you unfortunately got COVID, we can take your sera or blood or plasma, however you want to say it, and benchmark, is our vaccine doing better than natural infection? And at every step of the way, it's been doing equivalent or better, which is really amazing.
Yeah.
We don't have any data from the human trials yet, but so this is all just in mice and non-human primates, which we're comparing to humans. But
I see. And are those trials happening like in this area or?
No, I wish. So the phase 1/2 clinical trial is being run by SK Biosciences in South Korea.
Oh, okay.
And hopefully we will see data soon, but who knows when it'll be ready.
I'm sure you're eagerly anticipating getting some results.
Yes, I really want to know, is this something that can help people? And if it is, like, that's just incredible to know that I've played a small role.
Yeah, absolutely. So has this experience changed what you want to do moving forward as a scientist? Like, I mean, I'm sure you had ideas, back when you were getting your PhD about what your future would look like, but what are you excited to do now?
Yeah, I have changed, goals and ideas a number of times. So when I started grad school, actually the reason I came to grad school is I was really, and still am, really passionate about teaching and education. And that was really my driving force for going to grad school is, you need a PhD to be able to teach at the college level. And so that was really why I went. And then along the way, somehow I've really fallen in love with research and with the unknown and, figuring out burning questions that, before the pandemic, only a few people cared about, but I was one of those people who cared. So it was really fun and exciting. And so when I graduated, I was thinking, I'll stay on the research path, probably switch fields. I wasn't really attached to coronaviruses or to infectious disease, but I really just liked the questioning and the style of research. But I think now that I've really, sunk my teeth into the whole process, right, from ordering the gene to vaccine design, it's really exciting to think that, that I could do this for my career, for a living, that I could, bring things from research stage to the market. Like it's been really fun. It's been really exciting. It's been really rewarding. It's also been a lot of hard work, but that's normal.
Yeah. So I mean, so you have all this great experience now. Do you have people knocking on your door, asking if you want to come work for them?
No, but I do have, it's been really interesting to, reach out to my network and, see what's out there and what's open. And I have been, talking to people and thinking about my next steps, but it's also hard to move on when you have so many exciting things, going on. We don't just have this vaccine in clinical trials. We've also worked on, monoclonal antibody therapy. We have a lot of stuff in the pipeline. And as much as I'm ready to grow and move on, it's also still super exciting being here.
That's a good place to be.
It is, but it also, I feel torn in two directions sometimes.
Well, I think you shouldn't feel rushed to to move on. I mean, if it is going that well and you guys have so many good things going on, I think there's probably a lot of benefit to sticking around a little longer. Although I know the pressures of our career field.
Exactly. But, for now, I'm sticking with what's fun, and so far it's still fun.
Well, it's certainly, I mean, like how far you guys have taken this from ordering the genes like you said to phase 3, that's really incredible, and commend you for that, and all of your work on this disease and making advances. I think as someone who's worked in an adjacent field that's not infectious disease, it's definitely felt like, oh, I kind of wish I was working on, COVID and helping with everything that's been going on.
I can tell you all the downsides if you want, if that'll make you feel better.
Well, if you want to talk about it, yeah, let's hear them.
I think the hardest thing, is it's really hard to balance your own needs and time away from work when, especially early 2020, we needed answers so badly. And, I knew it, my boss knew it, literally everyone is asking you and waiting for your results. And there's a lot of pressure to create good quality science as fast as humanly possible. And that means, sacrificing your nights, your weekends, your mornings, your sleep, your everything, because to do it as fast as we needed to and as reproducibly, you have to be in lab a lot. And there's a lot riding on you. You and the small field of coronavirus experts who started in 2020, now the field is massive, which is incredible and amazing, but there weren't that many of us starting who, everyone was waiting for us to find what to do next.
What kind of hours were you pulling in those days?
That's not a fun question. There was a series of weeks to months where I was there till late at night. I would get home, sleep, wake up, and go right back. And there was no weekend, there was no free time. And, science takes a lot of planning, but I've never before planned my experiments like down to the hour per day for weeks out, because like experiment A had to work to set up experiment B, to set up experiment C, to repeat the whole thing, to make sure it worked again. Like it was a lot.
Yeah.
And this while the world was shutting down, and I was walking to work on dead, empty streets, and like my work life was very different from most people's work life in March of 2020.
Yeah. Was there a point where you were able to kind of take a break and, step away from the bench? And when was that?
Yes, we all, there's a huge team of us who are working on this, and we all needed to, share responsibilities so we could all stay happy and sane and, thinking of the next big idea and the next experiment that we wanted to do. But I would say, you know, by summer 2020, we were breathing again. You know, things had been moving, and you know, we had gotten kind of the first wave of really intense work out of the way. And kind of since then, it's come in waves. You know, you have really hectic 3 weeks and then like a slower down week, and then really hectic and then slower down week. And that's just kind of become the new pace.
Yeah. I'm curious, like, because I went back to lab in like April of 2020, not working on coronavirus,
Yeah.
but you know, I think there can be a similar urgency. What was your thought process like around the actual, like, you know, risk of you getting COVID, and you know, just walk us through that, like, over the course of time? And because, you know, I think it's like this interesting experience of, like, you know, going to lab and working on this thing and showing up every day, and also acknowledging that, like, you know, you're at risk yourself.
Yeah, no, absolutely. And it was a lot of emotions and a lot of roller coasters and a lot of, you know, you had to justify to yourself. You're like, is the work I'm doing, you know, worth me going to lab, right? Like the whole goal was, you know, if everyone just stays home, we can contain this. And it's like, well, I'm working on the thing we're trying to contain. Is that worth, you know, leaving my house and going and doing these things? And then, yeah, the other side is the self-preservation side, right? like, do I feel comfortable going to work and doing these things? And I don't know what this says about me, but I never questioned it. You know, I was like, this is the most important thing I could be doing. I'm going to be as safe as I possibly can. You know, I remember the first time that we started wearing masks at work, and it felt weird. And then I went to the grocery store, and, you know, no one in Seattle had a mask yet. And I wore my mask to the grocery store because in my head, I was like, I'm a danger to you all. You all have been home for the last 2 weeks and I'm the dangerous one. So I need to, like, and yeah, it was a weird time.
Yeah.
And even, you know, you're working late, and one of the things that, you know, you do when you're working late to get yourself home safely at the time was, you know, you take an Uber or you take a Lyft. And then it became a point where, well, that's actually not safe for me to do. So you have to balance, like, how late do you stay versus like getting yourself home safe, because you can't take an Uber because you can't be in contact with people, like all these logistical things that we took for granted before the pandemic, I think.
Yeah. Yeah, absolutely. And I think it's interesting because I think one of the aspects that was surprising to me was also, like, I think when you work in a lab, you become adjusted to protocols and, you know, safety practices, and wearing gloves and having a piece of plexiglass between you and your cells. Right. And for me, it felt like after, you know, going to work for months, it was like, well, as long as I kind of, you know, have my safety protocols, like, I'm not going to get sick, and that's fine. Yeah. And I feel very comfortable, like, out in the world. Whereas a lot of people who didn't have that experience and weren't going to work and were in their homes, like, had a lot more fear and, totally. You know, anxiety around, like, going out.
Totally. And I think everyone has gone through their own journey and their own roller coasters of, do I stay home? Can I go out? How do I feel about going out? Am I washing all my groceries? Am I not washing all my groceries? You know, like, we've gone through a lot of phases of pandemic as humans and as scientists.
Definitely. Did you know early on that, like, I mean, because obviously we weren't sure, a lot of people weren't sure about the, you know, infectivity from surfaces. So that was like a big concern. Did you know relatively early that it wasn't as big of an issue as?
Not any more than any other scientist or well-informed individual. You know, I remember on bioarchive when the surface paper came out, and, you know, I know some of the people who worked on it. And so, we all saw the paper and we were like, okay, it can survive on this, I forget now, but like plastics for 2 days and paper for 1 day, whatever it was. And, as scientists, we kind of take that and run with, it's data. And so I said, okay, plastics 2 days. So when I buy something plastic from the grocery store, it sits in a little box for 2 days and then we're good. But I think we also know as scientists that experiments are, they're done in a box or in a vacuum or whatever they're done in, and it's not the real world. It's not perfect. So no, I didn't know, but it doesn't surprise me. Like hindsight, it makes sense that it's different than what happens in the hood or in the lab or on your bench.
Or even from like a infection standpoint of a respiratory illness. Interesting. Yeah, I remember when that paper came out and it was disconcerting. It was like, oh God.
And then you're like, how many surfaces am I touching in a day? Like, how have I survived the last however many years of my life touching all of these things?
Right.
Thank goodness for immune systems. They are amazing.
Seriously. Okay, I have one other kind of nerdy question about how we measure like case positivity with the qPCR assay. Do you have any thoughts on that? And like has your thinking on that changed at all over time?
I am definitely not that is totally not my field of expertise. And I think, there's all this debate of, like PCR versus rapid and, how long are you positive after you're actually positive and when is a test too sensitive? And, I think these are all really important questions, but I think if I step back and I take, my biochemist, my very minute detail hat off, and I start thinking on a population level, in my opinion, the most important thing that we can know is, are you infectious or are you not infectious? And are you about to be infectious or are you not about to be infectious? And so, I think testing is super important, and there are definitely rights and wrongs or betters and worses, but in general, like, I'm very for getting all the tests that you possibly can.
Yeah.
But
like test agnostic, like any, I get
exactly. That. And, there's also, like we said, this pandemic has been extremely expensive, and there's, price and time turnover differences between the tests. And like, these are all things that we have to think about. But from a population level, I think all tests all the time are good.
Yeah, I did some digging into some of the like rapid tests as well. And I think, a few got approved here and there, but FDA kind of dragged its feet a little bit on some of them. And I think it could have been beneficial to have more rapid tests available to more people, at a certain point in time in the pandemic when it could have, allowed schools to reopen earlier and things like that.
Totally. Or, even just, especially schools are a great example, if everyone could take a rapid test, on their way into school, and then it's kind of a yes, no, it's much better than the temperature checks or the, daily attestations that people might've been doing. If you could literally get a yes, no every day, like people can feel a lot safer and a lot better about it. But, that's expensive and it's invasive, and, there's so many factors to think about. It's incredible what we've learned in the pandemic about not just the virus, but about humanity.
That's so true. So to kind of wrap up, we both live in Seattle.
Yeah.
We've, I think, fared relatively well through this pandemic, and the city's kind of reopening. Summer is here.
Yeah.
What are you looking forward to most about this year, either personally or professionally?
Yeah. So I actually just came back from a vacation where I got to hang out with my new niece. I'm a brand new aunt. So that was very, very exciting and one of the things I was looking forward to and hope to do more of in the future. But honestly, I'm a avid hiker. And so just the sunshine coming out again has given me some life and some energy and some excitement again. And so I'm just excited to be outside in the mountains, in the lakes, everywhere I can be that's not the lab when I have a break.
Totally. Do you have any favorite hikes?
Yeah, I think for Seattle natives, my favorite, doable hike is Gothic Basin. It's
Oh, yeah.
gorgeous. It is, if you're not from Seattle, it's just another world. Like the first time I did it, I'm not from Seattle originally, and it was just this fairy tale mountain land, and I loved it.
It's that one's incredible. I've done that one
Yeah.
a couple times and it's so awesome.
Yeah, and colors at sunset and sunrise, and just all this powerful rock surrounding you. It's, yeah,
Yeah.
out of
Have
this
you
world.
done the backpack up? Because I haven't. I've only done the hike.
I would definitely recommend the backpack.
Okay.
Just, spending a night up there. And also, there's a lot of other side hikes you can do off of Gothic. And yeah, it's
Isn't
incredible.
there's like an old mine back there or something
That
too?
could be true. I don't know that though.
Okay.
What's your favorite hike if you hike?
Um, last summer I had a chance to do El Dorado with
Hmm.
one of my coworkers who's a mountaineering guide.
Nice.
And we did the like Knife's Edge summit,
Nice.
which had been a goal of mine for a long time. And it was just probably the most amazing summit I've done in my
Nice.
life. So
I've not done that one, and I will put it on my list.
yeah, we did it in a day, which I would not recommend.
Good tip. Good tip.
I lost a couple toenails.
No.
But
But it was worth it.
it was worth it. It was really amazing. I mean, just like you summit, like it forms this snow edge.
Hmm.
And so you hike along the edge. And it's, I don't know, it's very surreal looking.
That sounds right up my alley.
Yeah, you should check it out. And it's in like the North Cascades, so you get views of like Glacier and all the
Nice.
surrounding mountains. It's really really an awesome view up there. So,
Nice.
but yeah, I haven't done too much mountaineering yet this year. So, got to get back out there.
Yeah, the season is upon us.
Yeah. Yeah. I've been actually working a lot in the lab. So,
No, I mean, yay, but no.
yeah. Yeah. But I mean, it comes in waves, like you said. And
Mhm.
I think when you've worked in this field long enough, you just get used to that. Like, oh yeah, I'm in like a work phase right now,
Yeah.
and I'm in the lab on the weekends, and
Yep. Yep.
then you kind of reset and get
Totally.
back. But
Totally. And, it's exciting. Like the stuff you're doing, the stuff like science is exciting. It's fun. That's why we work so much.
I know, yeah, actually, we were working. Me and my RA today, we were super busy in the lab, and then I just like looked at the results right before I had to come home to like get set up for this, and
Yep.
I got some good hits.
Yay!
So I was like, oh yay.
Worth it. It's worth it.
It was worth it. But yeah, is there anything else you want to touch on during the interview?
I don't think so. We touched on most of it.
Yeah. Yeah, I mean, if I missed anything, I think we got through all the questions that I had.
I think the only other thing that I usually, I try to say is, like we have four approved or three approved vaccines in the US. And, that's incredible and amazing. And hopefully we can, share these globally. But really the reason that this was able to happen is because so many people like me and before me and at the same time as me were working on really a lot of variety of things that, at the time there was no pandemic. No one cared about coronaviruses. No one cared about mRNA vaccines. No one cared about so many things that we now are looking at the news every day for. And, that's all because of public scientific funding. And it's not just funding the current pandemic, it's funding everything and anything, because you never know what the next important thing is.
Absolutely. Yeah. And I mean, I think one thing that's kind of unique and incredible about some of the research that happens here is that it really has allowed for us to make these types of breakthroughs and identify treatments that are so effective and life-changing. And I think, I mean, for many of us in the field, it's just such a privilege to be able to say, I'm fully vaccinated and I can go to work and not be worried about getting my coworkers sick or
totally
bringing something home to my family. And yeah, it's been, I mean, I think that's definitely been the light, at the end of the tunnel for a lot of us. So
yeah, that's what we're all, smiling into summer of 2021 with. It's hopefully a sore arm.
Yes. Well, I just want to say thank you for all the work that you've done on coronavirus and understanding these viruses, and then, pivoting and really working on developing novel vaccines. And I think, just the type of vaccine you guys have developed is really exciting, and I'm eager to see the data. And
me too.
I can't wait. Hopefully you can update me on whenever it comes out. And it's just been exciting to finally see that type of data. Like when the Pfizer paper first came out, it was just like unbelievable.
Yeah.
So yeah, good things ahead.
Yeah.
And thank you so much for joining us on the show and sharing all of your experiences.
Yeah, thank you so much for having me. This was awesome, and I really appreciate it.
Yeah, absolutely. That wraps up our interview with Lexi Walls. Wow, I learned so much about her work on coronaviruses and her experience working as a scientist during the pandemic, as well as some of the novel vaccines that she's been working on. It was great to chat with you, Lexi, and thank you all for listening to our show. If you can, subscribe, leave us a comment. We really appreciate the support. And thanks so much for listening. We'll see you next time.